Hello, and welcome back to the MCAT grind. We have Biology Chapter 2, Division and Reproduction. We have mitosis and meiosis, a cool, quick review. Mitosis, one cell divided into two identical cells. All of the genetic content will be exactly the same, except for, when things go awry. We have our G1 phase, S and G2 all grouped together as collectively called interphase. That is when the cell is not dividing, so that's 90% of the time. And the mitosis phase, which is 10% of the time, where it actually does the division. In G1 phase, there's increase in size of the cell, and between G1 and S, there is a checkpoint to check that DNA is not damaged. And then, if the DNA is not damaged, then the DNA will be replicating in the S phase, and then it will proceed to get bigger again. The cell get overall bigger to prepare for division in the G2 phase. And before going to the mitosis phase, the G2 will check that all of the DNA has successfully been replicated and there's adequate amounts of organelles and size to do the division. Then what we have here are the steps in mitosis. We have prophase, metaphase, anaphase, and telophase. Prophase will be condensing the chromosomes. Right now, during mitosis, all the chromosomes there's still 46 chromosomes, but there are 92 chromatids in total because each chromosome now it's like a little ribbon, right? It's connected like this. Then they will line up on the metaphase plate during metaphase, and then during anaphase it will be physically pulled apart at their kinetical cores by the spindle fibers generated from the centromeres on either side of the cell. And then in telophase, the cells will then split apart, and then finally cytokinesis will then be pinched off. And the chromosomes also basically unwind, so telophase and prophase are basically opposite of each other. For cells that don't divide, they will be arrested from G1 phase into G0 phase. And P53 is the important molecule or the chemical that's responsible for that arrest. And if P53 is mutated into TP53, I think it's called tumor promoting, that's what it stands for, and then the mitosis will be uncontrolled and you could have cancer. In meiosis, it's from one cell into 4 different cells. This is getting ready for sexual reproduction. So basically it goes through prophase, metaphase, anaphase, and telophase, everything 2 times to make 4 different cells in total. They are different because during the prophase one, they would have the mom and dad chromosome, analog chromosomes, the 23 pairs, so 46 chromosomes in total. They would go together in pairs and they would do crossing over events. When they cross over, they exchange some DNA material, and because they align all differently on a plate, the 23 pairs, we have the law of independent assortment, which is the second law of Mendelian genetics, which means that when they separate, you get so one set of genetics won't affect the other set. So you have basically two separate cells at this point. When meiosis 2 happens, you have the separation of the chromatids, and because one of the chromatid is from a native state and one has had a crossing over event, they will also be different and be pulled apart to different sides. And that is called law of segregation, and that's the first law of Mendelian genetics. What we have now will be the spermatogenesis. and oogenesis, and they're for meiosis. They're basically making from 46 chromosomes into 23 chromosomes, because after they split, they want to have half each of a set, and when they combine together to make the zygote, there are 46 chromosomes in total. Now let's look at the reproductive parts. In male, we have seminiferous tubules where the sperm are initially made and synthesized from spermatogonia. Spermatogonia is the primary part from which it's generated, and then later it will be stored in an epididymis, and it will also be matured there and added the flagellum. Sperm will be generated from spermatogonia into primary sperm, into secondary sperm, and then finally spermatozoa. During ejaculation, it will go from epididymis to vas deferens, ejaculated duct, into urethra, and then and then finally outside of the duct. Sertoli cells are present in the epididymis, or inside the testes, to help nourish and mature the sperm. And interstitial cells are generating the testosterone necessary for the production as well as the building of muscle parts in males. Seminal vesicles, prostate gland, and bulbourethral gland, the three of them help contribute to semen. Seminal vesicles provide the energy and glucose needed to power the sperm motility. Prostate gland add alkaline to ensure that it can bypass the acid part in the vaginal canal, and the bulbourethral gland generates the cleaning fluid to help clear out parts inside the urethra. In the female reproductive system, there are the vaginal canal, which the sperm goes through, and the main part is the ovary, where the egg is produced and stored. So all of the eggs are already produced when a female is born, and once per month, one egg will be continued and allowed from meiosis 1 to keep, continued from meiosis 1, but be arrested at metaphase 2, because they don't want to progress past metaphase 2, because remember, during metaphase, they are all lining up, and if they progress, they start splitting. They want to wait for the sperm to arrive and deliver all the DNA content before they start splitting into from a zygote and split into more cells. So all of the initial eggs are arrested at prophase, prophase 1, and then only once per month, one gets to keep going until metaphase 2. So in female, we have the menstrual cycle from follicular phase, ovulation phase, luteal, and menstruation phase. In follicular phase, the ovary is stimulated by the follicular stimulating hormone, and it stimulates the formation of a follicle. So what that means is it forms a little bump and allows the egg to mature to metaphase 2. Ovulation occurs when there's a luteal hormone surge. That surge causes the egg to be released from the follicle, and then from that remains of a follicle, it is generated into a corpus luteum. Corpus means body, luteum is luteal, so you can think of it as luteal hormone generating the luteal body, and that body is what generates progesterone. Progesterone is what maintains the uterine lining. So you can think of the uterus as the womb, and the lining gets thickened by estrogen initially, which makes the lining, and estrogen is produced by the ovary, and the progesterone is produced by the corpus luteum in the ovary after a follicle has been ruptured. and that maintains the thickness. If fertilization does occur, the zygote produces human corticotrophic, or HCG. I don't know, I forgot what it stands for, but HCG acts as the luteal hormone and stimulates and maintains the progesterone level. And since progesterone maintains the lining, that thickness gets maintained. Now, I have put them all together. We have initially follicle stimulating hormone, stimulated from hypothalamus initially in the brain, stimulates gonadotrophic releasing hormone, and that gonadotrophic releasing hormone acts on the anterior pituitary gland and allows it to release the follicular stimulating hormone, and that follicular stimulating hormone acts on the ovary to release estrogen. The LH hormone is also released from the anterior pituitary gland, and you can see their levels over time from 0 day to the 14th day, which they peak, and that peak, the LH surge peak, causes the egg to be released. Ovulation occurs on exactly the 14th day. Could be a 1 day less or 1 day more, but about there. And what happens is estrogen gets rise to a high level due to being stimulated by the FSH. There's an initial bump of the FSH, and that slowly increases the estrogen level. Then, due to the LH surge, the egg has been released, and the corpus luteum needs some time to form. Once it starts forming, there's a bit of a lag, and progesterone level starts to rise and be maintained. Now, once the progesterone level starts to fall and no fertilization occurs, the corpus luteum falls and sheds, and progesterone level decreases, so there's nothing to maintain the lining of the uterus, so that lining starts to shed and slosh away. And at the same time, in the beginning, estrogen will inhibit hypothalamus to prevent the releasing of another egg at the same time, so it does a negative feedback. So that is generally how division and reproduction goes. Good luck with the grind.